Hello, hello. For my topic, I decided to talk to you about action potential and its role and importance in the human body. Just a quick note: it says Delaney James, but my GCU account is still Delaney Naughton because my name has not yet been changed with the university. I just wanted to point that out. It's not someone else making the presentation. I'm just going by my new name. Okay, let's get into it. So, over the next 10 or so minutes, we are going to discuss what an action potential is, how an action potential is formed, and what processes in our body require an action potential. You might be surprised to know just how many times a day you need an action potential for your body to have basic function like moving or heart pumping and things like that. So, what actually is an action potential? Let's think about the physiology of wanting to move your thumb or even smile like our athlete over here. Oftentimes we just hold up our thumb and we don't think about why that happens. Um, in this case, there is a neuron that must cause your thumb to move. But how does the thumb know to move, whether to go up, left, right, down? In simple words, an action potential is a change in the electric charge in a cell. In complex terms, it is a rapid sequence of changes in the voltage across a semi-permeable membrane, that membrane that surrounds all of our cells. The change of ions and their concentrations that we're going to get is what's going to allow that action potential to actually start. I like to think about it in this way: the word action is referring to the outcome of the potential, meaning that there is the potential needed for an action to occur. Potential comes from this voltage in the membrane, which is determined by our ratio of ions, intracellular and extracellular, in and out of the cell, along with the ion's ability to permeate or pass through our cell's membrane. So basically, the action potential is the potential needed for your neurons to send a signal from your brain to different parts of your body to complete an action. That action requires potential from your cell. Action potentials are typically talked about in the context of a neuronal cell, but they are needed in other cells throughout your body. Your endocrine cells are sometimes used. Your muscle cells also need action potential. A command must be sent from your brain, so it starts in your brain and it travels down your spinal cord and it initiates an action potential at the beginning of a neuron located in your spinal cord. Once that command is sent, the action potential will travel down your neuron's axon and it's going to end up in the muscle that your brain wants to move. So, in our case of our smiling, thumbing up athlete, this action potential will travel down the neuron and end up at the muscle in your thumb, which tells your thumb to move, and so your thumb is going to move up. The action potential must travel from the cell body to the other end of the cell. So I mentioned some parts of a cell, things like the axon. I mentioned the spinal cord and stuff like that. You might be thinking big picture. Okay, we start in the brain, we travel down the spinal cord, we go through our body part like the arm, and we end up in our thumb. Let's think smaller picture. So, what actually is a neuron? A neuron consists of a main cell body called the soma with dendrites that are going to receive signals, these long branch like structures coming off of that main cell body. It also consists of a long axon or trunk covered in myelin sheaths that are going to actually help insulate the axon and help send signals to the other end of the cell. The opposite end of the cell is going to be called the axon terminal. So on one side we have the soma or the cell body with dendrites at the very end. We travel down this branch or this tube to get to the other side called the axon terminal. Direction of action potential is always going to travel from the soma to the terminal, carrying the action potential at the same strength the entire time. I like to think of a neuron as a tree. That's why there's a picture of a tree here. The roots are going to symbolize the dendrites, and where they meet could be considered the stoma or the soma. Roots in a tree are going to collect nutrients, and they send that nutrients up into through the trunk of a tree out to the leaves. Kind of like our dendrites are going to receive signals, send that signal up through the axon and out of the terminal. So our soma would be kind of like where all of our roots meet. Our dendrites would be all of our little root systems. Our tree trunk is going to be our axon, and our branches and leaves at the very top could be like the branches of the axon terminal. So now that we know what a axon looks like, we talked about the basic delivery of an action potential. Let's get into the nitty gritty or the actual physiology of what an action potential is. An action potential begins when the resting potential is usually around negative 70 millivolts, reaches a threshold level of about negative 50 millivolts. So we get positive. This step of the voltage changing is called depolarization. This first jump from negative 70 to negative 50, however, occurs a little differently. It's when neurotransmitters are going to bind to channels, and it's going to allow our positive ions, remember we went negative 70 to negative 50. So it's going to allow positive ions to trickle in, and it's going to make the cell slightly positive. This is going to occur at the soma, at the cell body, and once that threshold level is reached, typically around that negative 50, okay, we're going to see the action potential fire. This depolarization and the entire action potential is pretty much dependent on two main channels: a voltage gated sodium channel and a voltage gated potassium channel. So we have the threshold reached. The next point is really this rapid depolarization. And so what's going to happen is our voltage gated sodium channels are going to open, and sodium ions are going to rush into the cell very, very quickly. Those sodium ions carry a positive charge. So we have all these positives building up inside of our cell, and then it's going to rapidly depolarize. The cell is going to rapidly become very, very positive. So depolarization will take place until the membrane potential reaches about positive 40 millivolts. So we've got super duper positive super quickly, and our sodium channels are all along our axon. So what's going to happen is our axon is going to get a little bit positive at one point, and then that's going to kind of jump start the next set of sodium channels, and they're going to open and sodium's going to flood in, and that's going to jumpstart the next set, and they're going to open and sodium's going to flood in. So we get this domino effect occurring down the axon of our sodium channels opening and sodium flooding in. So as quickly as our cell is depolarized, it gets super duper positive, it's going to get repolarized. So our peak potential is about positive 30 to 40 millivolts, as I stated at the last slide. And once that is reached, our voltage gated potassium channels are going to open. And now positive potassium is going to move down its concentration gradient and out of the cell, removing our positive charge of the cell. So at the same time our potassium channels open and lets potassium out, our sodium channels are going to close so no more sodium gets in. So voltage gated sodium channels are open, sodium floods into the cell and makes it positive. It reaches a certain potential until we let the potassium channel open, which lets potassium out. And the whole goal is to restore our concentration gradient. However, oftentimes our cell will overdo itself. Too much potassium floods out and the cell becomes too negative, and the action potential is pushed too far. This is called hyperpolarization. And in order to restore the resting potential, something called the sodium potassium pump is going to bring the cell back to its original concentration in order to prepare the cell to fire another action potential. The signal is then automatically propagated, which means to spread to the next set of channels throughout the axon. I like to think of it as a stick of dynamite or maybe a sparkler on the 4th of July. You light the top of a fuse or the or what are they called? You know what I'm trying to say. You light the top of the fuse, and that spark is going to slowly travel down that fuse until it reaches the end of the stick of dynamite when you get that reaction. Same thing with our action potential. It's going to start in our soma and our cell body, and it's going to propagate, spread step by step by step through the entire axon until we get to the axon terminal. Because if our brain wants to send a signal, we don't want a weak signal by the time it reaches our axon terminal. We want that signal to be as strong as it was when the brain first sent it. So here's an image of a very well-known graph depicting membrane potential in a neuron that is firing an action potential. We see this horizontal line staying constant at negative 70 millivolts, that blue line, and a slight depolarization is going to occur to reach that threshold, which is that green dotted line. Once again, this occurs from those neurotransmitters binding and allowing just a few positive ions into the cell. That threshold is reached, our sodium channels open, sodium floods into the cell, and that is represented by a very steep blue slope here, one increasing. Repolarization is going to happen to restore the membrane's potential, and our potassium channel is going to enter. Potassium is going to flood out. But remember, our cell is going to overdo it sometimes. So we see this hyperpolarization, that little dip in the graph there. To remedy this problem, our sodium potassium pump is going to transfer sodium potassium against its concentration gradient using ATP until we can get back to that resting potential. This is going to repeat itself once again until that action potential reaches our targeted muscle that we need to receive the signal. So our refractory period, and in this slide I'm also just going to close everything up and talk about why we need action potentials. So an action potential is an all or none basis, which means that an action potential is either going to happen or it's not. It would be kind of compared to a water fountain in a school hallway. The water is either going to come out or it's not. You either press the button and you get water, or you press the button and you don't get water. There's really no adjusting the amount of water that comes out. A neuron is going to send the same size action potential each time. Either we reach that threshold and we get the action potential, or we don't, and we don't see an action potential at all. Excuse me. So, when the neuron is going to send an action potential, it really can't control the size of the action potential. So, how do we know if something is really important? So, the neuron must adjust our frequency of the action potential to get this important information received faster, or maybe something super important, it needs to happen really fast. Think if your hand touched a hot stove, you didn't know the burner was on. Your brain really wants to send a signal to your hand to move your hand super fast in order to reduce the damage to your skin cells or your muscle cells or your nerve cells on your hand, because that's what's going to happen. Your brain senses, oh no, I really need to get that hand moved. Let me send a signal down super fast, and then it's going to move. So, how fast the brain sends signal is going to indicate the strength. For example, a stronger signal will have a higher action potential frequency. Frequency, then, is determined by refractory periods. There are two types: absolute refractory and relative refractory periods. In an absolute refractory period, the cell will not be able to send another action potential at all. It is absolutely impossible, because sodium channels are going to stay locked shut for a period of time, meaning no sodium can pass through, which will make it impossible for our cell to reach that threshold to send an action potential. This does, however, help the cell direct an action potential down the axon. In a relative refractory period, it is harder to send an action potential, but it's not impossible. It's going to occur after this absolute refractory period, and our sodium channels are no longer locked, meaning sodium has the ability to flood into the cell. But our cell is going to be in hyperpolarization, meaning it is further down the negative than we want to be, than our resting potential. So sodium has to send larger concentrations into that cell in order to reach the threshold to send another action potential. So we learned that action potentials are this rapid change in charge that allows neurons to send signals, and we focused on the process that an action potential starts and being sent down the neuron. We've even explored Very briefly, what a neuron is, its shape, and its structure in order to better understand action potential propagation. Now that we have a basic understanding of action potentials, they are a great importance of our body. Action potentials are needed for the brain to function because they're responsible for sending information to the nervous system, to the central nervous system, and to propagate commands initiated in the central nervous system to the peripheral nervous system. Things like the spinal cord and your brain from the central nervous system to the nerves in your arms and your legs and your toes and your thumbs and all of those things to move your body. If action potentials were not an all or nothing, our movement would be very, very drastic, would be almost like a spectrum. Um, but being able to send an action potential is what's going to allow our body to respond to our environment. Here are my sources. I really hope that you enjoyed this presentation. I'm sorry if I rushed it. I'm working on it. But I hope you have a blessed rest of your day. Thank you for listening to me.